Hello friends, welcome to Biogam. We'll be talking about worst medical disaster in the history of mankind. Thalidomide. In a short while, we will be able to realize how this episode has taught us many valuable lessons. Thalidomide was first synthesized in 1954 by a German company Chemicoriental, chemically known as alpha-N-thalamidoglutaramide. This drug was prescribed for a number of conditions, including flu, as an antiemetic, and for causing sleep. People referred to this drug as miracle drug. This drug gained immense popularity and was marketed in various countries under 52 different trade names. This drug was even made available without prescription at some places. Chemicoriental claimed thalidomide to be a side effect-free drug. They claimed that the only side effect to this drug would be the induction of deep sleep. Thalidomide gained immense popularity shortly after its introduction, but then something went wrong. Though thalidomide was claimed to be side effect free, the claim did not sustain. There have been various reports of nerve damage and deformed babies. Company at first ignored these reports as there was no direct link and kept reassuring about the safety of the drugs. However, the reports of side effects kept growing, and it was only in 1961, after Lenz and McBride independently suggested the link, and the drug was withdrawn. However, the damage was done. It is impossible to estimate the number of victims of thalidomide, but more than 10,000 babies were affected. The adults were also affected by peripheral neuritis and memory loss, but a generation of children suffered a huge, irreparable loss. Thalidomide is a synthetic derivative of glutamic acid. Glutamic acid is a naturally occurring amino acid involved in important physiological processes. Research on thalidomide further revealed that it has a chiral carbon which is unstable. The unstable chiral carbon has been marked with a circle in the thalidomide structure. The result of instability of the chiral carbon is the coexistence of two enantiomers. It was found that R form was responsible for the claims which the company made. However, due to spontaneous racemarization, R form was converted into S form in human blood. This S form was responsible for the side effects. The drug was sold as a racemic mixture, as isolation and administration of a single enantiomer was not possible due to spontaneous racemarization under physiological conditions. Research further revealed the critical exposure periods of thalidomide. This figure provides critical exposure periods for thalidomide embryopathy during human development. Note that the critical exposure periods overlap with the period when pregnant women begin to experience morning sickness and nausea, for which the drug was prescribed. A diverse collection of mechanisms of action of thalidomide for teratogenicity has been proposed. Major mechanisms are oxidative stress, DNA intercalation, inhibition of angiogenesis, and cerebellum binding. Other mechanisms include acylation of macromolecules, ascorbic acid synthesis, decrease in adhesion receptors, and cytokine synthesis. The proposed mechanisms are not mutually exclusive. It is quite possible that several of the proposed mechanisms are working in parallel or synergistically to result in teratogenicity induced by thalidomide. According to Greenthal, the manufacturer of thalidomide, the testing of the drug was carried out using the standards of the time. It remains a matter of debate as to precisely what testing was carried out and when Greenthal became aware of thalidomide being a human teratogen, whether the disaster could have been prevented any sooner. However, this disaster completely changed the way drugs are tested. Thalidomide disaster first demonstrated that species differences exist in drug reaction or response. As in this case, mice, which was traditionally used to screen for drug action, are less sensitive to thalidomide than other species like non-human primates and rabbits. Drug screening policies have changed to incorporate several species as well as in vitro tests. This disaster has also shown us the importance of absolute chiral specificity in biological systems. The packaging and information of contraindications of drugs was minimal when compared to the drugs being sold today. This disaster resulted in improved and more robust parameters for drug testing. After a period of ban, thalidomide's potential was rediscovered. Dr. Jacob prescribed the drug as a sedative to a leprosy patient on compassionate grounds in 1964. He observed an unexpected and dramatic relief of the patient's lepra skin within 48 hours of drug administration. This finding was later reproduced and reported by him in 1965. In 1971, World Health Organization published the results of a randomized trial that confirmed the efficacy of thalidomide in treating leprosy. Nowadays, it is being used to treat a number of conditions including leprosy, multiple myelomas, and cancers. Its uses are due to its analgesic, anti-inflammatory, and anti-angiogenic properties. Now the drug is being marketed only after being subject to robust clinical trials. Thalidomide use is being carefully monitored using successful schemes like STEPS, which stands for System for Thalidomide Education and Prescribing Safety Programs. Such schemes monitor patients on a day-to-day basis and ensure that patients do not make or get pregnant while receiving treatment or during the treatment. Also, the sharing of thalidomide without proper prescription is prohibited. Many organizations, such as Thalidomide Victims Association of Canada, have come out with education and awareness campaigns to make people understand the risks involved. Thanks for watching. Please like and subscribe Biogan.